In this figure, we present evidence that parvalbumin-expressing basket cells differentiate between CA1 pyramidal cells that project to different brain areas. First, we injected fluorescent microspheres into distinct brain areas and found retrogradely labeled pyramidal cells in the deep sublayer of the ventral hippocampus. When we injected red beads into the medial prefrontal cortex and green beads into the amygdala, we found that the majority of the pyramidal cells were labeled only with one of the two tracers, indicating that the same pyramidal cell rarely projects to both areas. Next, we performed paired patch clamp recordings from hippocampal slices. Action potentials in the presynaptic basket cells evoked larger inhibitory currents in pyramidal cells that projected to the amygdala compared to those that projected to the prefrontal cortex. This was a key result that demonstrated that these interneurons do not provide a homogeneous or blanket inhibition, but are in fact highly selective in terms of the pyramidal cells that they innervate. Were the pyramidal cells themselves also selective? Indeed they were. When we tested the connections in the opposite direction, we found that medial prefrontal cortex projecting pyramidal cells were much more likely to display excitatory connections with the basket cells than their amygdala projecting counterparts. This is a schematic representation of our data illustrating a highly biased inhibitory and excitatory microcircuit. This circuit is organized in such a way that when the hippocampus sends information to the medial prefrontal cortex, it preferentially excites the basket cells and thus inhibits the output to the amygdala.